Tiny hunters, big hope. Could bacteriophages save humanity? Somewhere in a drop of ocean water, an invisible war is happening. Not between sharks and fish. Not even between bacteria and your immune system. But between bacteria and something even smaller. Meet the bacteriophage, a virus that infects and destroys bacteria. Or, if we're being dramatic, and we are, the microscopic predator that might one day save us all. The most abundant assassins on Earth. Let's start with a mind-blowing fact. There are more bacteriophages on Earth than every other living organism combined. Yes, really. Scientists estimate there are over 10 to the 31st power of them. That's a number so big it makes your math teacher nervous. If you lined them up end to end, they could stretch hundreds of millions of light years across space, far beyond our galaxy. They exist everywhere, in oceans, soil, rivers, even inside your body. Anywhere bacteria live, phages are there too, quietly hunting. Despite their alien, robot-like appearance, some look like tiny lunar landers. They have one simple job: find bacteria, inject their DNA, take over, destroy. No drama, no mercy, just efficiency. How a bacteriophage attacks like a sci-fi movie. Imagine a bacteriophage landing on a bacterium like a spaceship docking. It doesn't swallow the bacterium. It doesn't enter it fully. Instead, it does something even cooler. It injects its genetic material like a syringe. Once inside, the bacterium becomes a factory. Not for itself, but for the enemy. Within minutes, it's producing dozens of new phages. And then boom, the bacterium bursts open, releasing a swarm of new attackers. This is called the lytic cycle. And it's basically a microscopic version of a zombie outbreak. But not all phages are this aggressive. Some choose patience. They quietly insert their DNA into the bacterium's genome and lie dormant. Sometimes for generations before activating. This is known as the lysogenic cycle. A kind of biological sleeper agent strategy that makes phages even more mysterious. A forgotten discovery, rediscovered. Bacteriophages were discovered over 100 years ago. In 1917, scientist Felix Darel realized that something invisible was killing bacteria. And even used it to treat disease. For a moment, it seemed like humanity had found a miracle cure. But then came antibiotics. Simple, mass-producible, easy to prescribe. Phages? Complicated, unpredictable, and not well understood at the time. So the world chose antibiotics and largely forgot about phages. Except not everywhere. In places like Georgia and parts of Eastern Europe, phage therapy quietly continued for decades. Like a backup plan waiting in the shadows. The problem: antibiotics are failing. Here's where things get serious. Bacteria are evolving fast. Every time we use antibiotics, we give bacteria a chance to adapt. Some survive. Those survivors become stronger. Eventually, we get superbugs, bacteria that resist multiple drugs. Some predictions suggest that by 2050, drug-resistant infections could kill more people than cancer. That's not a sci-fi disaster. That's a slow, quiet crisis already happening. So the question becomes, what happens when antibiotics stop working? Enter the phages again. This is where bacteriophages make their dramatic comeback. Unlike antibiotics, which act like a bomb, killing both harmful and helpful bacteria, phages are snipers. They target specific bacteria and leave everything else alone. That means your beneficial gut bacteria stay safe. Side effects are reduced. Treatment becomes highly precise. It's like switching from a flamethrower to a guided missile. Even more interesting, phages don't have to replace antibiotics. They can work alongside them. In some cases, bacteria that evolve resistance to antibiotics become more vulnerable to phages and vice versa. Scientists call this phage antibiotic synergy. And it could become a powerful strategy in future medicine. Real-life miracle stories. This isn't just theory. It's already happening. One of the most famous modern examples is Tom Patterson, a psychiatry professor at UC San Diego. In November 2015, Patterson contracted a highly virulent superbug. While vacationing in Egypt, the bacterium, Acinetobacter baumannii, sometimes called Iraqibacter, was resistant to nearly every antibiotic available, including last resort drugs. His condition rapidly worsened, and he fell into a deep coma. With no conventional treatments left, his wife, epidemiologist Stephanie Strathdy, launched a global search for help. Doctors eventually turned to bacteriophages as a last resort. Under a special compassionate use approval, they assembled a personalized cocktail of phages, viruses designed to target his exact infection, and administered it intravenously. Within 3 days, Patterson woke up. Over the following months, the infection was brought under control, and he eventually returned home after a long recovery. Let that sink in. A virus saved a human life. Not just medicine, phages everywhere. Phages aren't only used in hospitals. They're already quietly working in everyday life. Food safety. Phages are sprayed on foods to kill harmful bacteria like listeria. Agriculture. Protect crops from bacterial disease. Water monitoring. Detect contamination. Hospitals. Used to sterilize surfaces and equipment. There's even a product that uses phages to clean food safely. Yes, a virus you can spray on your salad. Sounds scary, until you realize it only targets harmful bacteria. The catch, because there's always one. Before we declare phages the heroes of humanity, let's be honest. They're not perfect. 1. Too specific. Phages target only certain bacteria. That's great for precision, but tricky for treatment. Doctors must find the right phage for each infection, often by screening large phage libraries. 2. Bacteria fight back. Just like they evolve resistance to antibiotics, bacteria can also develop resistance to phages. But this battle goes both ways. Bacteria have even evolved systems like CRISPR, a genetic defense mechanism, originally designed to recognize and destroy invading phage DNA, long before humans adapted it for gene editing. 3. Delivery and regulation challenges. Phages are larger and more complex than typical drugs, making them harder to store and standardize. And there's another problem. Phages are living, evolving entities. Because they are biological rather than fixed chemicals, they are often treated as living drugs, which makes standard clinical trials far more complex. As a result, they don't fit easily into traditional drug approval systems, which are designed for stable, unchanging substances. A strange but beautiful partnership. Here's something poetic. Bacteriophages don't attack humans. They can't infect our cells at all. Yet they live inside us, constantly shaping the bacteria in our bodies. In a way, they're silent guardians, keeping harmful bacteria in check without us even noticing. So, are they humanity's future? Maybe. Or maybe they're part of the future. The truth is, bacteriophages aren't here to replace antibiotics entirely. They're here to work alongside them. A future treatment might look like this. Antibiotics weaken the bacteria. Phages target the resistant ones. Together, they win. Kind of like a tag team battle against invisible enemies. Final thought, the smallest hope. It's funny, isn't it? For decades, we feared viruses. We blamed them for disease, pandemics, and global chaos. And now, one type of virus might help save us. Bacteriophages remind us of something important. Sometimes, the solution to a big problem is hiding in something incredibly small. So next time you think about the future of medicine, don't just imagine giant machines or new drugs. Imagine a tiny, spider-like virus, quietly hunting bacteria, and maybe, just maybe, protecting us all.
It's time to study new words in context. 1. Predator. Definition: an animal or living thing that hunts and eats other living things. Example: the lion is a predator that hunts smaller animals. 2. Abundant. Definition: something that exists in very large amounts. Example, water is abundant in the ocean. 3. Swarm. Definition, a large group of insects or small animals moving together. Example, a swarm of bees flew around the tree. 4. Lytic cycle. Definition, a process where a virus enters a cell, makes many copies of itself, and then breaks the cell open. Example, in the lytic cycle, the virus destroys the bacteria after making copies. 5. Genome. Definition, all the genetic information inside a living thing. Example, scientists study the human genome to understand diseases. 6. Dormant. Definition, not active, but still alive and able to become active later. Example, the virus stayed dormant in the body for many years. 7. Lysogenic cycle. Definition, a process where a virus hides inside a cell and does not destroy it right away. Example, in the lysogenic cycle, the virus stays hidden in the bacteria. 8. Adapt. Definition, to change in order to survive in a new situation. Example, animals must adapt to changes in their environment. 9. Vulnerable. Definition, easy to harm or attack. Example, without protection, the body is vulnerable to infection. 10. Phage antibiotic synergy. Definition, when bacteriophages and antibiotics work together to kill bacteria better. Example, phage antibiotic synergy can help treat strong infections. 11. Virulent. Definition, very strong and harmful, especially for diseases. Example, the virulent bacteria made the patient very sick. 12. Epidemiologist. Definition, a scientist who studies how diseases spread and affect people. Example, the epidemiologist studied how the virus spread in the city. 13. Intravenously. Definition, into a vein in the body, usually using a needle. Example, the medicine was given intravenously in the hospital. 14. Sink in. Definition, to be fully understood after some time. Example, it took a moment for the news to sink in. 15. In check. Definition, under control and not allowed to grow or spread. Example, the medicine kept the infection in check. 16. Tag team. Definition, two or more people working together to achieve something. Example, the doctors worked as a tag team to treat the patient.
Comprehension questions. 1. What is a bacteriophage? Answer, a bacteriophage is a virus that infects and destroys bacteria. 2. Where can bacteriophages be found? Answer, they can be found everywhere, such as in oceans, soil, rivers, and even inside the human body. 3. Why are bacteriophages called predators? Answer, because they attack, take over, and destroy bacteria. 4. What happens during the lytic cycle? Answer, the virus enters a bacterium, makes copies of itself, and then breaks the cell open. 5. What is the lysogenic cycle? Answer, it is when a virus hides inside the bacterium's DNA and stays inactive before becoming active later. 6. Why were bacteriophages forgotten after their discovery? Answer, because antibiotics were easier to use, produce, and understand. 7. What is the main problem with antibiotics today? Answer, some bacteria have become resistant, creating dangerous superbugs. 8. What are superbugs? Answer, bacteria that can survive many or all antibiotics. 9. How are phages different from antibiotics? Answer, phages target specific bacteria, while antibiotics kill many types of bacteria, including helpful ones. 10. What is phage antibiotic synergy? Answer, it is when phages and antibiotics work together to kill bacteria more effectively. 11. Who was Tom Patterson? Answer, a patient who was saved from a deadly infection using bacteriophage therapy. 12. What made Tom Patterson's infection so dangerous? Answer, the bacteria causing his infection were resistant to all available antibiotics. 13. Why is phage therapy difficult to use widely? Answer, because phages are specific, must be matched to each infection, and are hard to regulate as living drugs. 14. What is CRISPR and why is it important? Answer, CRISPR is a defense system used by bacteria to fight viruses, and it is now used in gene editing. 15. Why are bacteriophages considered a hope for the future? Answer: because they may help treat infections that antibiotics can no longer cure.